let's quickly check from the notes also. A transcription unit consists of 3 regions in the DNA. These are promoter, structural gene, and the terminator. Remember, this arrow plays a very important role, so don't forget to put, because this shows the direction of the reading and the point from where transcription starts. The 2 strands of DNA in the structural gene forms a transcription unit. As 2 strands have opposite polarity, an RNA polymerase, DNA-dependent RNA polymerase, also catalyze polymerization in one direction. Therefore, DNA strand that has the polarity 3 to 5 act as a template and is called as the template strand. The other strand, which has the polarity 5' to 3' towards the promoter end, and with the sequence same like RNA, except it will have thymine at the place of uracil. I hope this is clear. Otherwise, please send me a message in the WhatsApp group. I will explain again in the next video. Is called as the coding strand. All the reference points while defining the transcription unit is made with the help of the coding strand. Promoter and terminator region of a transcription unit are located at the either ends of the structural gene. Promoter region is present towards the upstream. Now please remember, upstream means 5' end and downstream means the 3' end. Like that means, if you remember this diagram, the moment you say, because the reference has to be taken with the coding strand. So when you say promoter is present upstream, upstream means towards the 5' end. So you can see here, when a statement in the book says that the promoter is situated upstream of a transcription unit, that means upstream means 5' end of the coding strand. And when it says the terminator is present towards the downstream of the structural gene, so downstream means 3' end, and 3' end of which one? Coding strand. So every reference is made with the help of the coding strand. Now the next point is, references are made with the help of the polarity of the coding strand. It's a rule you have to remember. Terminator region defines the end of transcription. So transcription will start from promoter and then will end with the terminator. The presence of a promoter in a transcription unit defines the template and the coding strand. Switching the promoter with the terminator, so if you change their position, would reverse the definition of coding and the template strand. A number of regulatory proteins are also present upstream and downstream. We'll be learning about that in next pages of the NCERT. Now the next thing to understand is the transcription unit and the topic of genes. Now the term gene was coined by Johansen. We have already covered this in 5th chapter, to replace the term which was given by Mendel, and that term was factor. He defined genes as an elementary unit of inheritance which can be assigned to a particular character. Now Mendel stated that these factors are the genetic information, something which is controlling the inheritance and expression of a particular character. This term was replaced by Johansen, was replaced by gene. So according to him, gene is the elementary unit of inheritance which codes for a particular character and controls the expression and controls the inheritance of that particular character to the next generation. Morgan work on the crossing over and recombination suggested that gene is a segment of chromosome which can be separated through crossing over and is therefore a unit of recombination. Now there is a confusion among scientists because gene initially became a very popular word, but slowly its definition became very tough, because as biology advanced, it became tough to give a particular one-line statement of genes, because initially this was the statement given, but after Morgan's experiment it became clear genes are able to undergo crossing over. So if you remember that crossing over concept, we have already done that. Recombinase enzyme will cause the crossing over and genes may undergo separation. So according to Morgan, what is gene? It's the smallest segment of DNA which can undergo recombination. So the other name given by a scientist called Benzer is recon. So a smallest segment of DNA which is able to undergo or which is able to separate during recombination is called as recon by him. Similarly, the work on mutation suggests that gene is the smallest segment of the chromosome which is able to undergo mutation also. So Benzer, that's why I called it as the muton. So what is gene? Gene is the smallest segment of DNA which is able to undergo mutation, and the new term introduced by him was muton. So now genes are able to control inheritance, genes are able to undergo recombination, genes are able to undergo mutation. So that's why factor, gene, recon, muton, even replicon is there. The smallest segment of the DNA which is able to undergo replication is called as replicon. So many words were introduced that it became tough which definition should be the best one for genes. That's why a new term has already come in biology, and that is called as cistern. I will be talking about that. Now after this, what happened? Beadle and Totem, two scientists, they recognized the gene. They performed experiments regarding one gene, one enzyme, or one gene, one polypeptide hypothesis. So Beadle and Totem recognized a gene to be a unit of heredity that contains information for the synthesis of enzymes. And Janowski and his group in 1965 observed that a gene has information for a single polypeptide. However, DNA not only contains information for synthesis of single polypeptide, but also carries information for formation of other RNA like tRNA, rRNA. So that's why so many terms have come into existence. So the new term now which has replaced the genes is cistern. It can be defined as a segment of DNA capable of directing the synthesis of polypeptide chain through mRNA or any type of RNA molecule. A segment of DNA capable of forming a chain of amino acids through messenger RNA, or which codes for formation of other types of RNA like tRNA and rRNA, would be called as cistern.